All right, I think I see Tom's clock has turned to 1:02, so we'll get started. I want to thank you all for being here today. We had a tremendous response for registration from folks all over the world, so we're super excited to have you here. I'll do a brief introduction of myself and what we're doing here today. My name is Jeffrey Seitz. I'm the Vice President of DMark Biosciences. And we are a genomics or omics solution provider. We're now almost 18 years old, a Canadian-focused solution provider, and we generally serve the academic research community in Canada. We're excited to bring to you today one of our partners, Inventia Life Sciences, and this is the latest in 3D cell culturing, and very exciting stuff. I want to say that part of what we do at DMark is try to bring you not only these amazing technology and new tools, but also education and, trying to bring you tools beyond just the research that you're doing every day. I'm going to do a plug here for DMark. We have a couple of events that we are co-hosting with academic institutes coming up. These are full-day symposiums called GEXI Life Course at UBC on May 8th, and we're hosting another event, GEXI event at McMaster on June 8th. And so, sorry, of course, the dog barks during the webinar. So anyway, I'm going to pass it off because of that, but I thank you again for attending here today, and I'll pass it off to Craig.
Hey, everybody. Thanks very much for joining. Thanks very much, Jeff. Certainly a pleasure to have the opportunity to speak with you today. Jeff mentioned my name, Craig Powell. I am the distribution manager here at Inventia Life Science. And part of my role is to support partners such as DMark as they share news and information about our technology with investigators such as yourselves. I will emphasize that Inventia Life Science is an Australian-based company founded in 2011 with offices in the US and also in the UK. Now, at its core, Inventia is really a company of talented engineers and biologists that have developed a solution to overcome many of the issues and challenges that are encountered in generating, reproducible 3D cell culture models. And to introduce the technology and the platform is my colleague, Dr. Tom Grundy. And Tom holds a PhD in cellular and molecular biology from the University of Sydney, and he's been with Inventia just on 6 years. Also joining us today are Whitney and Hannah from Inventia Life Science, and they will help answer any questions that you can or that may pop up in the chat. So without further ado, I'll hand it over to Tom.
Thanks, Craig. Yeah, it's great to be here with everyone today. And yeah, I'm going to be talking and introducing the technology a little bit, but my main focus here is case studies, talking about use cases, how people are using this technology, and the biology surrounding it. And what I'm hoping for is maybe a bit of a discussion as well, if people have thoughts, challenges about working in 3D with current model systems or systems that they're thinking of using. We're more than happy to have that discussion and talk those challenges and possible solutions through. So I know the majority of the people in this webinar today have an interest in pancreatic and islet research. And the challenges and requirements in this area of research, there might be more, and I'd be happy to hear from the room if there are more. But some of the challenges include the fact that you're working with sensitive cells, often patient-derived samples. You're necessarily needing to create complex multicellular models. Maybe this involves endothelial and islet cells. Maybe you need to also explore immune interactions for your in vitro models. And you need this kind of basis for your model, but you also then need the preservation of key phenotypic features. And for a good in vitro assay, you also need consistency and reproducibility, and to a degree, throughput, whatever that means for you. Ultimately, it's great to have a model in a well, but to kind of take it on and use it for downstream analysis, you'd need that consistency and that reproducibility well to well, and you need to be able to generate that at a high enough throughput and with good enough viability to get meaningful results. So that's kind of the big picture of the challenges. Obviously, there is relevance for these models in various kind of stages of research, whether it be early disease research, early discovery, preclinical work, and all in all, we like to think of this problem space as something that requires a phenotypically relevant model. And you might hear me use that term a little bit during this presentation. So for us, what does that mean? A phenotypically relevant model, it exhibits a clinically relevant phenotype. That could be a physiological phenotype, or it could be a disease phenotype. That's going to be characterized by a variety of different features. Maybe not all the disease features are key to your research, but there will be select ones. It might be a particular marker expression. It might be a disease morphology, a disease characteristic, but you need a preservation of those in your model. And then, as I said, you want it to be standardized, reproducible, scalable, and compatible with a variety of different downstream analysis strategies. And so that's what we're going to be talking about today. We unfortunately don't have many case studies that directly relate to pancreas and islet research. We've got some work in pancreatic cancer that's been done with our system that I'm going to talk about. We do have a lot of examples, though, that are going to touch on these general challenges and requirements. And hopefully, after speaking with you guys, we can have ongoing conversations and we can really start developing some complex 3D models in this space. So I'd also like to touch on this kind of classic association between scalability and complexity. So typically, as you have a more complex model, you lose scalability, you lose throughput. And you can think of that with some examples that I've put up here. So, you've got simple spheroid models on one end. You can start making those models more complex, adding more cells, adding ECM elements. And then you can also start to add physiologically relevant elements to that. So it might be flow, like fluidics, compartmentalization, things like that to your model system. But typically, as the complexity goes up, you lose throughput and therefore you lose scalability. What we're seeking to do with our system is to get the best of both worlds, allow you to get that complex model that you need for good research, but do it in a scalable and even high throughput manner. So I'm going to offer a really brief technical overview of our system. I'm not going to dive too deeply into this because, as I said, I'm more interested in talking about the case studies. But obviously, some people are new to the technology, and so a brief overview is probably required. So big picture, what we're offering here is a matrix or matrix options that is a synthetic, they're based on a synthetic backbone that's been functionalized in a way that supports phenotypically relevant cell behavior. But because they're synthetic, that gives you a lot of experimental control as well. We're taking that matrix, which is supporting the cells and the development of those biologically relevant models, and we're pairing it with our hardware. So that's allowing us to create these consistent and reproducible models in standard in vitro formats. So 384, 96, 24 plate options, depending on your research question or your particular downstream analysis strategy. And then putting this together, what you end up with is the ability to create reproducible disease models so that they're clinically relevant. And as I've said, consistent, reproducible, and easily analyzable. So a little bit more information on our matrix. We have a library of matrix options. So essentially whatever cell line you bring through the door, whether it be patient-derived, primary, iPSC-derived, continuously cultured, where we have matrix options that can support those cell lines from whatever tissue origin as well. Essentially, we have a PEG matrix backbone that we've functionalized with cell adhesion peptides, MMP degradable ligands, and in some instances, full-length extracellular molecules. This allows us to control the stiffness of the matrix, but you're getting all this complexity in a synthetic product, so we also get no batch-to-batch variability. You're going to get the same results time after time. And that GIF in the middle is just showing what happens when we add our cell recovery fluid to the matrix. So, you can leave the cells in the matrix and do a lot of work with them in, but if you're doing an analysis strategy that requires retrieval of those cells, we have a solution that can allow you to retrieve those cells and go on with your workflows. And that's been used in some of the case studies that I'm going to present. So this is like an overview of what a standard workflow would look like. The first two steps, the matrix selection and the model architecture selection, that can be done either using our software, our cloud-based software, which is free, or it can be done in consultation with someone like myself or Whitney or Hannah, which I guess is also free. So at the matrix selection stage, either like myself or someone else or the software would be helping you select what matrix specificities you need to support the model that you're trying to create. The next step is selecting a model architecture, and that's going to be based upon your research questions. So maybe you're doing an imaging-based analysis, and so you'd go for an imaging model. Maybe you need more cells, so maybe you go for a large plug model. Both of which print in a 96-well plate, or maybe you need a screening model because you're doing something a bit more high throughput. In any case, there's a variety of different model options. So once you've done that, it's on to the lab-based part. Preparing your cells, it's essentially a 2D cell culture workflow. You're mixing your cells in with one of our reagents, just like you would resuspend cells in media before plating them up. Instead of plating them up, you're putting them into our cartridge. The cartridge goes straight into the Rastrum, or in this case, the Rastrum Allegro, and you're off to printing. And then everything downstream is essentially, the handling is very similar to how you would handle a plate in 2D. So really it's a 2D workflow before and after printing, but you're getting 3D results. And I've actually put two case studies in this initial section because I think together they really demonstrate quite nicely the power and potential of our offering. So this first case study, this is a case study. This is some work that was completed by BMS. It was presented at a conference, and we have permission to share it. So that's pretty rare and quite nice. And essentially what they were looking to do is create accurate phenotypically relevant models of pancreatic ductal adenocarcinoma, so PDAC, which is just a nasty form of cancer, as I'm sure a lot of people on this webinar would know. They were doing this using patient-derived PDAC organoids. In some cases, they were doing it in a co-culture with patient-derived cancer-associated fibroblasts, or CAFs, and then they were obviously combining it with our matrix. So essentially, like to gloss over the workflow, they were taking these cultures, they were printing them into plates, culturing them for amount of time, treating them with drugs, both some control solutions and some novel compounds that they were interested in testing, and then they were running them through endpoint analysis. So just looking at this central part of the slide here, You can see at the top, a big part of what they were interested in testing out here, BMS, and also what I think is a takeaway from this slide, they were seeking consistency between plates, within plates, and between different model sizes that they were looking to use. So you can see this first culture was printed in a 96-well plate, then they printed in a 384-well plate. So they were looking for consistency between those conditions, and that's what they saw, which they were obviously happy with. Obviously, when they added the calves into these cultures, they saw a change in morphology. That's not surprising. I mean, what really did stand out to me in this case, though, was how impactful that addition of that extra cell line was in terms of gross cell morphology. And then they went on to complete some viability testing, both using live dead stains and cell-titer glow. Again, what they were looking for here was both intraplate and interplate consistency. And as you can see, the results they got were pretty stunning. So, I mean, there you've got a 96-well and a 384-well plate model. It's looking pretty consistent again, excellent viability. And then they got very consistent results. So day 0 CV, so really just a test of like, without biology taking its course, what's the starting CV? Intraplate CVs of around 7% in this 96-well imaging model format, and around sort of 12% with this 384-well screening model. So they were really excited about that. And then, of course, they went on to complete their drug testing. They were excited because, again, what they saw was an alignment of those IC50s and those drug results with their internal data, but now they're using a system that's allowing them to create these models. A 96-well plate in 2 minutes, a screening model in 4 minutes. So the potential for then completing a screen using these more complex 3D models is something that they're now looking to move forward with. So yeah, to go back to why I'm presenting this slide, I think there's some detail there, but big picture, I really think it highlights the consistency and reproducibility of our system, the ability and the compatibility with high throughput workflows. On the other end of the spectrum is the use of our system to create these more complex models, but still do so in a consistent and reproducible fashion. So this is some internal work that we did. We were interested in creating a tumor stromal interface and a model that recapitulated that. And so what we did here was print this triple matrix model. Even though in this schematic you've got green, pink, and yellow segments, we put the same culture, this same co-culture of endothelial cells, primary endothelial cells and primary fibroblasts in the top green and bottom yellow sections. And then in the middle section here, we included A549 lung adenocarcinoma cells in co-culture with those same primary fibroblasts. What was really exciting for us about this was you did see the development of these more mature endothelial structures in this endothelial compartment. So you can see here, the CD31 staining, you're getting these endothelial structures developing, and then they're abutting to this tumor compartment. And there is this overlapping interface. So we are getting a really nice in vitro model of this tumor stromal interface. But because of the printer, we're able to create that interface in an XY plane as opposed to a Z plane. And that's advantageous for high content analysis and just generally downstream analysis, imaging-based analysis strategies. So yeah, I think this really nicely, again, summarizes the offering here for people interested in creating complex models. You can take something something that's quite complex from a biological, in vivo setting. You can model it nicely in vitro, and you can do so in a highly consistent and reproducible way that then facilitates simplified downstream analysis. So that's the introduction. I'm going to go on now and talk through a few different case studies. I actually can't see whether we've got any questions. So maybe if we do have questions and they're in need of answering, maybe someone can just let me know. So this section is focusing on sensitive cells and patient samples. You can probably see as I go through this that a few of these case studies could probably be put in different sections and are relevant to different kind of general focuses, but I just thought to organize them in this way. So this first case study is some work completed by a group in Australia. And what they were interested in doing was taking patient-derived organoids that were generated from pediatric tumors, patient-derived pediatric tumor samples, so neuroblastoma, and a couple of different kinds of sarcoma, so osteosarcoma and Ewing sarcoma. They were taking these samples, culturing them in 3D, also in animal models. And then ultimately they were interested in completing a drug screen, which is what they did in the bottom left here. But initially what they were really concerned about, kind of, I guess, like BMS was, are the models that we're creating relevant to the original samples, or have we got genetic shift? Have we seen a change in cell behavior or disease characteristics? Are our models relevant to the original sample? And we're happy to say that that is what they saw. Sorry, that wasn't what they saw. They didn't get a change. So they printed these models and then compared them back to the original samples, and what they saw was there was a preservation of key molecular and disease characteristics in the printed models when compared to the original tissue samples. In terms of any genetic drift, they were pretty happy as well. They saw a goodness of fit of 95.3%. And then in terms of biological behavior, cell behavior, they also saw consistency in that when compared to the original samples. So that was really exciting. And then obviously based off this, they completed a drug screen and saw some interesting results that they're now going to take on and explore further. So that example, I suppose, related a little bit more to patient-derived samples. Obviously, there's challenge, as always, in working with patient-derived samples. This one relates more to working with sensitive cell lines, and in this case, primary human hepatocytes. So, as I'm sure many people on this call know, that's a challenging cell line to work with. It can be hard to get long-term cultures. And we thought this was quite interesting in the sense that without a model that can last long enough, you may miss key biological or pharmacological interactions that would be relevant to take on or at least understand before moving on in a study. And so to kind of assay or explore this, we took triglyceridone. It's a drug that has been taken off the market, but it was known to cause chronic cytotoxicity, hepatotoxicity issues. And what we saw was, treatment with triglyceridone in over 2 days, you don't really get much of a cytotoxic effect. However, using our system, we were able to create these beautiful 14-day primary human hepatocyte models. I'm not showing all the data here, but essentially a lot of those key markers that you want to see with primary human hepatocytes were still going up at day 14. And it meant that we could complete a 7-day treatment with triglyceridone and expose this chronic cytotoxicity that the drug can cause. So yeah, I think that really, to me, highlights the benefits of our system in terms of supporting longer term cultures, even of sensitive cell lines such as primary human hepatocytes, and then the benefits that can be derived from that purely in terms of the model. So I've actually got two slides from this group. This was some initial work that they did, and then they've since taken it on, and I'll talk a bit about that later on. This work's actually being completed by Merck MSD in the UK. And again, it's published, so we can talk about it. But what they were looking to do here was derive long-term neural cultures. And the reason behind this is going to become more clear on the other case study slide that I'll present. But yeah, that was the challenge for them. But they saw really nice success with our system. So essentially, they did was print cells, complete a neural differentiation process of those cells that took 30 days, and then culture them for another 60 days. So all in all, the cells were in culture for 90 days. Our printed matrices held up, and they got phenotypically relevant cell behavior that really excited them and then spurred them to continue with the work and ultimately develop a bit of a disease model, which I can show in this next case study slide. What really excited them was the ability to cut down that differentiation protocol a little bit because they got these neural progenitor cells in 3D culture, which supported the cells in a phenotypically relevant way and allowed those cultures to mature much faster. And then obviously the gels are synthetic. They didn't get a background signal, so it meant like imaging-based analysis was very straightforward. They did some kind of high content-based analysis and they got really nice results. Just stopping there. Again, if there are any questions that I haven't seen, please let me know someone. That I'm assuming there's not. So this next section, I think I've only got 2 case studies in this section, and it's really a focus on phenotypic preservation, both disease and physiological, I suppose. So this first case study, it's some work that was completed at the Royal College of Surgeons in Ireland by Dr. Marie McElroy. And Marie's lab, among other things, they were working with patient-derived breast cancer samples. They'd had success previously culturing these samples in 3D using BME matrix options. However, what they saw when they did this was a loss of key disease phenotypes. Specifically, this luminal breast cancer subtype would grow, typically grows in this clumped phenotype, whereas lobular breast cancer cells grow in this sort of elongated single cell file phenotype. When they cultured these using BME options, the cells grew happily, but they lost this disease feature. Excitingly, what they saw when they printed cells using our matrix and rastrum was a preservation of this disease phenotype. So in the case of luminal breast cancer cells, you can see they're growing in this clumped phenotype, and that matched with histological sections. And similarly, this single cell phenotype matched up with what they were seeing in histopathological sections of that cancer. So that was pretty exciting. This slide, I mean, it touches upon, a preservation of a phenotype, that being a fibroblasts, activated fibroblasts, and polarized fibroblasts. But I think what this slide also nicely highlights is the compatibility of our technology with downstream analysis. So if anyone has worked with fibroblasts before and other BME options, they can be challenging to work with. If you're trying to keep cells in an unactivated phenotype, that can be challenging. If you're maybe introducing immune cells or macrophages to that culture as well, keeping everything in an unactivated state is a big challenge. But also, a lot of BME options are so soft and don't allow the cells to interact the right way that you don't actually get, a polarized fibroblast phenotype, which is pretty characteristic of those cells in vivo. With our matrix, the matrix keeps the cells in a quiescent state. So obviously they can be activated into a profibrotic state, but that's not their baseline in our matrix. And also you get morphology that's more characteristic of how those cells behave in vivo. In this work, we were looking to introduce an activated phenotype in them, and so the cells were stimulated with TGF-beta. And this panel here shows collagen 1 staining of those cells in our matrix compared to a BME option. So obviously you don't have control of the matrix elements in BME, and so if you go in and try and stain for an ECM component, you're going to have trouble. Oh, and we also did some gene expression analysis. So here I'm going to touch on complex models. And so this really touches a little bit more on that second case study slide I showed at the top. In fact, I might have removed it. But this other focus where people maybe are less interested in developing complex 3D models that are compatible with high throughput strategies, and maybe more interested in developing complex environments that are still consistent and reproducible for study of biological niches or interactions of different biological or pathological compartments. And I'm going to start here with some work that was completed internally on the creation of an immuno-oncology model. This work has now been continued on, or picked up and furthered by an academic group who has one of our systems in Australia. But it's kind of a nice place to start with the work that we did internally. So what we were looking to do here was create a simple immuno-oncology model. We took A549 lung cancer cells and embedded them in a printed matrix. This matrix actually has, this schematic is simplified. We actually print an inert layer underneath the matrix to ensure that anything within that printed matrix, one, cannot escape the matrix and form a monolayer, and two, cannot sense that underlying stiffness of the plastic surface and start changing its cell behavior based off that, which wouldn't be phenotypically relevant. So we print the inert layer, print this structure on top, and then after a few days in culture at a media change, we've added in T cells. And what we're looking to see here was that the T cells would be able to infiltrate that printed matrix, migrate to those developing cancer cell structures, and then target and eliminate those cells. And that's what we saw. So this is obviously an IO example, but I do know in the pancreatic and islet space, there are groups interested in not only creating a model in a well, but then introducing immune cell elements and seeing how those cells interact with the model. And so I think this is highly relevant to this talk. You can see over time that the cells not only migrate through the matrix and start homing in on the cancer structures, but those cells are obviously secreting MMPs. The matrix is an MMP degradable matrix. So those migration paths are going to widen and more cells over time flood through them. So I said, that's sort of the basic version that we did internally. and explored internally. And this was the more advanced work that has now been completed by a group at the Garvan Institute in Sydney. So what they were looking to do was create a phenotypically relevant PDAC model system. And so what they were using was a KPC cell line that was overexpressing an over 1 albumin antigen. And then they were looking looking to target those cell lines expressing that antigen with OT1 T cells specific to the over 1 antigen. And that's what they did. Essentially, like the base model was quite similar. They were printing the PDAC cells in the matrix. They were introducing the T cells at a later time point. But I think just what's really beautiful about this is the work they did validating the model in comparison to a BME option that they were also exploring. And then the downstream work, the high content analysis, all of that, it's pretty spectacular. So there was a lot of validation work that they completed of the different components of this model that they were putting together, and I haven't presented all of that here. The one I did present, though, was just this comparison of their wild type T cells and how you really didn't get the homing in of those T cells to the relevant tissue in animal models compared to the OT1 T cells specific to that over 1 antigen. And I like to sort of set that comparison up because I think it nicely kind of compares to what they then saw in BME models when compared to our matrix. So you can see here, they embedded their PDAC cells in a BME matrix. First of all, they saw a very kind of consistent morphology that wasn't really relevant to the way that the disease presents in vivo. But then when they introduced the T cells, they saw that they just didn't get invasion and interaction of those T cells with the PDAC cells embedded in their matrix. Comparatively, with our matrix, they saw what made them a little more exciting in terms of cell phenotype or like multicellular phenotype, relevant disease markers, those sorts of things. But they also saw invasion of the T cells into the matrix, interaction of those cells with the developing PDAC structures, and ultimately killing behavior, which they assessed using cleave caspase-3. So yeah, all in all, I think this slide to me highlights the potential for creating complex models, to be creating models that are phenotypically relevant, and then to be able to, take those models, implement relatively complex downstream analysis strategies, and get consistent and reproducible data. So previously I referenced a group from Merck MSD in the UK who had worked on putting together a 90-day neural model. And this is the work that they then, it's a preprint currently, but they're looking to get it published, and it's pretty exciting. So they were taking that fundamental work and then they looked to extend it in the development of a Alzheimer's model. And what they were doing here was printing a co-culture of iPSC-derived neurons. They created like a complex forebrain neural culture, a mix of a variety of different cell lines. They introduced microglia right at the end. They had astrocytes, they had astrocytes in there, and then they also had Alzheimer's APP protein as like a I'm sorry, I just had a mental blank. But essentially, they had a disease model and they had a physiological model that they could compare between. And what they were looking to see was how those cell lines developed neural networks, and or neuronal networks, I should say. So what they did was print our triple matrix model. So this was the same model that was used previously, that we used to investigate tumor stromal interactions. In this case, they've printed in the top and bottom compartments, but they've left this middle compartment blank. So it's a blank shell that's been printed, and then they look to see the neurons develop and grow neurites into that, and the two populations interact with each other. They did see that successfully. They got nice bridging. They used different stiffnesses of matrices in different compartments to ensure that the soma of those neurites stayed in the top and bottom compartments while the neurites projected into that central compartment. And then they completed a bunch of really beautiful analysis on all of this. So assay-based readouts, IF, high content. They did some protein readouts of amyloid B, and then bulk RNA analysis as well. So yeah, we're really excited about this work. It's one of the first published instances of one of our more complex models being used, and the body of analysis that they were able to achieve using this complex model is pretty remarkable. So I think the final section that I'm going to be talking on today is downstream integration. And I only have a couple of slides, so there's time for questions or comments if anyone has any. So this first one, I thought I should put something in about a drug treatment, because I mean, I talked about drug treatment early on with the BMS work. But one of the questions we often get is, is your matrix compatible with treatment with bulky drugs? Or, I mean, I've already showed that we can get T cells invading in, but, I know that ADCs, antibody drug conjugates, are becoming more popular in research spheres. And so I thought this was relevant. Essentially, this work was done with like a co-treatment of PARP inhibitors and some ADCs. The group was just treating a continuously cultured cell line, so nothing too fancy on that side. But what was really exciting for them was that they saw good penetration of the drug into the matrix and good effect. This is only early work, but it was quite exciting, and they were just thrilled that they had a model that could reproduce the 3D biology that was necessary for them and support a drug treatment kind of like they were hoping that they would typically see in vivo. And this work, I think the main focus here is they've actually gone on and done a little bit more work in the same space. I don't think I'm not presenting on that today. But the thing that I really got excited about here, I mean, first, obviously, they're working with a sensitive cell line. They're able to get an improved differentiation protocol using that sensitive cell line, deriving mature cardiomyocyte cultures in vitro. And that was really exciting. They were able to reduce the time it took to get that mature culture. Again, they believe it related to the fact that the matrix was supporting those cells in a phenotypically relevant manner. But then their downstream readout was single cell RNA-seq. So this was the first group that wanted to use that analysis strategy with our platform. And they got really successful results. So they got, sequencing quality RNA out of their samples using that cell retrieval process. So, after these structures developed, adding in a reagent, extracting those cells from the wells, and then running them through a single cell RNA-seq protocol. So that was really exciting. And those are my case studies for today. But yeah, thank you. And are there any questions?
There's one in the chat here, Tom, from
Okay.
that Hannah's addressing, but it's basically asking if there's any work that you could address in diabetes.
Yeah. So, I mean, I think the reason that we're here and talking to you guys today is because we feel that that space has not been explored so much currently with our technology. We feel that what we have seen in terms of supporting sensitive cell lines, complex cultures, that sort of thing, is highly relevant. I think we could certainly take one of our matrix options and take some islet cells, whether they be patient derived or a primary line, and get those cultures going for you for sure.
Joe, there's no other questions currently. I'll ask one more time. Does anybody would like to ask a question of Tom
I
before
see
we?
there's one that says, how do you choose the right matrix for 3D culture? Any concern of artificial behavior? From Justin. That's certainly a challenge, Justin. I think the key factor here is what I advise to people is, you know, what is key for your experimental question or your particular disease? What are the factors that you need to preserve? And then starting from there, okay, let's find a matrix that best supports that. We've worked with enough people now, enough different groups that have been generous enough to let us put at least the matrix option that we used or advise them to use for their particular cell type or tissue origin in a database. And so we can use that to help select a matrix option for a novel cell line. And what we typically find, even with the most difficult cell lines, is it only takes 1 or 2 iterations, 1 or 2 plates, 3 matrix options per plate to work out a matrix option that's suitable for the cells. As I said, and as I presented, we're working with plenty of sensitive cell lines, patient-derived, primary, whatever. I mean, I've been at the company 6 years now, and I can't think of a cell line that someone's brought through the door that we haven't been able to find a matrix that supports it in a way that is, relevant to their research questions. So yeah, it's a pretty good track record.
Couple new questions there in questions and answers, Tom.
Oh yeah. So the one from Tuan, is that the one you're talking about?
Can you click on the Q&A on the bottom? I can read them out to you.
All right, let me see the Q&A. Can you talk a little bit more about the cell recovery process and what that entails? Yeah, sure. So it's pretty straightforward, to be honest. We have a matrix. We've got this synthetic matrix. Obviously, it's supporting the cells in this phenotypically relevant manner that I've been talking about. But because it's synthetic, we can also break it down quite easily with the right addition of a reagent. So we have a reagent, it's gentle on cells. We just add it to the matrix. It's a short incubation. Depending on the matrix stiffness, I think sometimes we suggest gentle agitation. And then you're just pipetting those cells out, and you've got your cells. You've got them in a supernatant, you've got to separate them out from that reagent, and then you can go on with your downstream analysis. So it's really straightforward, it's quite quick. I mean, our technology baseline is being designed by biologists for biologists, and a big part of biology is viability, right? You can't really do nice work with a non-viable sample. So that's really front of mind whenever we're creating cultures or working on products. Rebecca, Can you talk a little bit more about logistics of sending primary patient-derived cells for you to test in your system? So to demo test the system at a site where we are generating islets. We could certainly speak to you about that, or like at least have that conversation. We also do have a discovery services offering, and so if you were really concerned, like of seeing some validation work prior to investing in the platform, that's also something that we could support. Definitely, like there's a lot of different ways that we can make that work. Definitely come and have a chat to us. We'd be more than happy to have a chat.
We're
I'm
going
also
back to the
getting,
chat here, Tom. I'll read one off to you.
sure.
It says, for scientists that have familiarity with Matrigel, for example, what are the biggest changes and advantages that have seen with using your matrix option? How many choices of matrix do they need to go through to
Mm.
optimize their cultures?
Sure, that's a good question. I'll address the second part first. In terms of matrix selection, if you've got a cell line that we've worked with before and that database is rapidly expanding, it might just be something where we're like, okay, this is the matrix we advise, and it should work. Worst case, if it's something we haven't worked with, or so if you're going to bring in some primary islet cells or something, I would probably be starting with a matrix selection panel of 3 matrices. I'd expect one of them to work. If they didn't, it might take another iteration. We might try something else. And obviously we would be testing the validity. I mean, often people start with like a live dead viability stain, but then, with islet cells, we might also be looking for particular, secretory markers and things like that. But that's where I would be starting. And, I wouldn't expect it to be much harder than that. In terms of familiarity with Matrigel, what are the biggest changes and advantages? I mean, first of all, you're getting something that is controlled. You don't have to worry about batch-to-batch variability. You're controlling what's in there. I mean, I showed examples before of ECM staining. If you go and stain something for collagen 1 in Matrigel, the whole thing lights up. With our matrix, you're not going to get that. The ability to control stiffness, the compatibility with downstream workflows, and then just ease of interaction as well. I mean, anyone here who's worked with Matrigel knows that it's not a lot of fun to work with manually in a hood. Whereas with our system, you're getting these consistent reproducible structures. The models are better. The Matrigel domes aren't going to fly away. They're not going to break down over time. I mean, I presented that Merck work. They did 90-day cultures and the matrices were still there and still solid. So yeah, I think biological relevancy and ease of use, and then compatibility with downstream workflow. I think it's all relevant, and you'll get benefits across the board using our matrix compared to Matrigel.
Hannah and
So
Whitney are doing a great job in the
they
webinar.
are,
So
they're
it's
killing
kind
it.
of like back and forth, some questions on Matrigel viability. Things
Yeah,
you're both answering the same questions kind of in the live and in the chat.
yeah, right. Yeah, so again, touching on the viability part, we would be very confident in getting highly viable cultures out the other end. That I think we like worked with like Huvex. Actually, I can't remember. Essentially, the testing that we did internally, we validated viability with highly sensitive cell lines and got, industry-leading viability results from them. We get really solid results with primary human hepatocytes, with printed iPSCs. Neural cultures are, a classic one to kind of keep, which are hard to kind of keep happy and viable, and we've had great our customers and us, we've had great results with those. So yeah, in terms of cell viability, we're very confident in our platform and being able to offer you something that is going to match up at least in line or better than whatever benchmark you have.
There's a note here I want to read out loud from Hannah that I think is a nice one. It says, also great to know that images, as you've seen, are great with our matrices as they are synthetic. There's no autofluorescence. So that's a nice feature of the
Absolutely.
synthetic matrice.
We have a couple more.
Okay.
When releasing cells from a co-culture with a couple matrix, can you selectively release one matrix only while leaving the matrix intact? There is short answer, no, and that is one of the challenges when developing complex 3D models. And I think it's something probably good to just talk about generally anyway. I speak to people who are interested in putting 5, 6, 7 different kinds of cells in the same well, having them in different compartments, different co-cultures. Practically speaking, we can do it. However, the challenge is you add complexity to a model is you've got to keep everything happy, right? So we've got to find a matrix that works for all the cell lines in a select compartment. And if you have 7 cell lines in a single well, well, now we need a single cell media that's going to keep everything happy. So, that's often a balance with creating these more complex cultures. Adding complexity, maintaining control, because you obviously want control in an in vitro format, but also then managing that complexity with other factors. If you needed to release cells from a compartment of that, well, I think there would be ways that we could explore designing the model a little differently that would give you the same result, but make it more technically feasible. If if you needed them in the same well, I mean, one way that we could do it, for instance, would be print a larger version of that multi-matrix model in a 24-well plate, print it on a cover slip, and then remove that, look to separate one section from the other physically, and then complete the downstream analysis on it. So like there's kind of clunky ways that I can imagine that we could do it, and then maybe there's more kind of finesse ways that we could do it through experimental design.
Yeah, and my backup on that is that one requires an offline call to do
Yeah,
some more in-depth question
yeah,
and answering. So if you're
absolutely.
interested in exploring that, just let us know and we can dive into some technical discussion beyond Tom's answer here.
Yeah, absolutely. And I'm always
There's
keen
a
for
good
those
one
discussions.
here. What would be your final pitch to the CITRN group?
Is
I
that is
like the
that from me or is
question.
that from you, Jeff?
No, that's not from me.
That's from me. All right. I mean, I think my pitch would be that I think we have enough. I think we have enough case studies now that we've demonstrated a high level of compatibility with whatever's come through the door, and people are happy and excited and getting good results. One area we've done a fair bit of work in PDAC, as you can see, but physiological pancreatic models work in the diabetes space that hasn't been explored. I think that's a really exciting opportunity. You know, we pride ourselves on not just like dumping the box on your bench and leaving. We pride ourselves in offering ongoing support. And I'm very confident that if we had people interested in that space, that we could support you guys, get you some really exciting results, and yeah, help to create some really exciting models and generate exciting data, and ideally, solve some tricky disease problems and answer some disease questions. So yeah, I don't know. That wasn't a very kind of succinct pitch, but yeah, I think that's what my takeaway would be. Where all the pieces are in place. Yeah, we'd love to talk to you.
Well, we're over the hour now, and we have an empty box. It looks like we have the Q&A answered. So we'll call it. We'll make this a call, and thank you, Whitney, for your answers today. Thank you, Bhavya, who is the marketing manager at DMark Biosciences, for organizing this whole thing. There's quite a lot of effort that goes into organizing one of these webinars, so I want to make a special thanks to Bhavya for her help with this. Very successful today. Thanks, Tom, for going through the case studies in particular. And I welcome all of the attendees to reach out to us. We will all be following up with you individually, I'm sure. But please, if you have other questions, we're happy to answer those. We're happy to get on a call, as I discussed earlier with Tom's last question. So we're happy to answer more questions if you have them. Thank you again for your time today. And I wish you all a good afternoon.
Cheers. Thank you, everyone.
Bye-bye.